Hi, my name's Rachel Gregoire. I'd like to thank the organisers for inviting me to present today at this fantastic workshop, and also thank the technical team for being patient with my lack of IT skills. Today, we're going to be thinking about how we can improve implantation potential and clinical outcomes from ART. Specifically, I've been asked to talk to you today about how we can improve implantation potential when we look at oocyte and embryo quality. I have received an honorarium for this presentation, and I have no other further disclosures to make today. What I hope we can all take home and take back to work today and really think about is can we and how can we improve oocyte competency and quality? And we need to think about pre-egg collection for IVF, and also furthermore when we produce in the laboratory, when we have our eggs, our oocytes in the laboratory. Once we receive our oocytes, we need to think about how in the laboratory we can actually maintain the quality of those oocytes when we receive them. And when we do create fertilization and we have embryos that are in the laboratory, how can we maintain quality but also select embryos that have the best implantation potential? And we need to think about those different aspects of IVF, and hopefully from a laboratory perspective today. So why me? What do I know? And why have you asked me to present today? Well, I am not an academic or a researcher, so I'm hopefully going to give you something to think about from a clinical setting, from an IVF clinical setting. I'm a clinical embryologist. I have been for many years. I now have the position of scientific director at the Hewitt Fertility Centres in Knutsford and Liverpool in the UK. Our clinics are classed as large clinics. We do about 1,600 fresh cycles and about 1,600 frozen cycles across both sites. I also hold the position of what's known as the person responsible. So that's the person who's responsible to the UK government, where I have to ensure that we are compliant with UK law. I also sit on the executive committee for the Association of Clinical and Reproductive Scientists, or ARCS, as deputy chair, with a specific interest in education of our reproductive scientists of the future. So I want to take it back a step and think about natural conception first of all, because I think it's quite important, because what obviously natural conception does is it optimizes the environment of the oocyte and the embryo. And the laboratory and our stimulation and so on need to try and mimic this as best as we can to hopefully continue to optimize the potential of the oocytes and embryos that we create. So if you think about natural conception, we know that the sperm is deposited at the cervix. We know it migrates up towards the fallopian tube, where there is hopefully a competent oocyte that has migrated, that has been ovulated and is migrating down the fallopian tube towards the sperm. Now, whenever you read the textbooks on natural conception, I always think of it almost as like a nirvana, the ideal environment, and that you talk about gentle wafting of ciliated cells, the natural flow of secretions, these gentle contractions. It almost sounds like a spa treatment, and all of this is in either, it could be a chemical gradient or a temperature gradient to attract the sperm and the eggs together, sperm and egg together. And then the constant migration, hopefully, of the fertilized egg into the endometrium to hopefully implant around 5 or 6 days post-fertilization. So we need to look at this and consider what we're doing in ART, which is so different and really couldn't be further away from nirvana when we think of it from an egg and an embryo perspective. So if we think about ovarian stimulation, first of all, and I am not a medic, I am a scientist, so you will need to bear with me. We know, if we think about the natural ovulatory cycle, we know that the body is invested in that production of that one lead follicle. And we know that follicular function is there to support the development of a mature and competent oocyte. Now, naturally, follicular development is asynchronous because we have multiple follicles starting to develop, but one takes the lead and the others invest all their energy in ensuring that dominant follicle continues to grow while they start to degenerate, the others start to degenerate and fall back. Controlled ovarian stimulation is the same in that we have that asynchronous development, but what we don't have is that regression of the less competent follicles. So when we think about follicular function and think about the development of asynchronous follicles, knowing that there's follicles' role is that bidirectional communication with the thecal cells and the granulosa cells of the egg to ensure that it becomes competent. We can't help but assume that this asynchronous follicular development is going to lead to a combination of competent oocytes. So when we think about ovarian stimulation and the impact that it will have on oocyte quality, we think about that, well, follicular function is to develop a competent oocyte. A competent oocyte should be able to fertilize. It should be able to divide. It should be able to create a blastocyst. It should be able to implant. It should be able to create a viable pregnancy. So why are so many oocytes not competent? Why are so many embryos poor developing? And why do so many embryos not implant? And I think we do have to continue to look at ovarian stimulation and see what we can do to improve the impact it has on oocyte competence. And I'm going to leave that there. So then we think about the patient sitting before us just before they start their IVF cycle, and they want to know, is there anything they can do to improve that follicular environment? Again, you look at the literature, there are multiple publications on supplements that patients can take in the 2 to 3 to 4 months building up to IVF. Now, I'm giving an example here. In no way am I endorsing these supplements, and I'm just wanting to illustrate really that the dearth of evidence that's there, that we don't have robust data to support supplementation is going to improve oocyte quality, but I just wanted to give examples here today. So we take coenzyme Q10, for example. It's a natural molecule. We know that it protects cell membranes. We know that it creates oxidative balance. And we know that increased oxidative stress will lead to more mitochondrial and organelle damage. So there is some evidence to suggest that 2 to 3 months of coenzyme Q10 supplementation, pre-stimulation, will give an increased oocyte competence. It's safe, it's well tolerated by the patient, but the data to support that it does improve oocyte competence is poor. I've taken a study here in the last bullet point, which I think was only 60 patients, and it quoted improved morphokinetics. And I think already this makes me feel uncomfortable in what is an improved morphokinetic. It quotes one cell cycle in particular, which has no link to embryo quality, and the evidence that there isn't the evidence to support that this morphokinetic in particular, this cell cycle will lead to a better, more competent oocyte. And it also talked about slightly improved pregnancy rates. So the data isn't there to support the supplementation will improve oocyte quality, but the patient wants to do what they can, and it's well tolerated and it's safe. Is this something that we could say, well, there is no evidence, but it can do no harm if you want to try something? We then have the consideration of DHEA. And again, when you look at the literature, there is literature there supporting the use of DHEA pre-stimulation. It's an endogenous steroid. We know it's already around in the ovarian thecal cells. We know it's a pro-hormone for testosterone and estradiol production. We know it can reduce apoptosis and cell death. And again, is there an argument that this could, 3 to 4 months of supplementation, lead to improved oocyte quality. Again, picking out one paper in particular, which is cited several times, many times in other papers, is that there is a suggestion that DHEA can improve oocyte and embryo quality. This was a study on 5 patients. So you can see again that there is a lack of robust data supporting that anything can improve quality of oocytes before we reach egg collection. But we need to be prepared for these conversations and be honest with our patients when they're in clinic. So we have our oocytes and we have our stimulated patients, and they are ready to have their egg collection, and the eggs are going to come into the laboratory. We're talking about things that we don't really know the right answer of in vivo improvement of oocyte quality. But let's think about in vitro. We know some things. And this is where we need to really ensure that our laboratories are safe and are maintaining the quality of the oocyte as it reaches the lab. What we do know is that temperature has a significant impact on the quality of the oocyte. With irreversible cooling, with irreversible damage done by cooling eggs to around room temperature, 23 degrees. It will break down the spindle. It will then reduce fertilization and create poor blastocyst development, and will increase genetic abnormalities in those oocytes because of the irreversible damage. This is a paper from way back in 1995, but it's kind of a key paper that we always come back to. And this demonstrated that if an oocyte was exposed to room temperature for up to 2 minutes, it could be reversed if the oocyte was returned to 37 degrees incubation. If it wasn't, that was irreversible. If it was exposed to room temperature for more than 2 minutes, there were signs of irreversible damage, even if you did then incubate the oocytes. And this is really important for laboratory procedures in IVF, because there are many times that a dish is left out of the incubator while we perform our laboratory techniques. So what can we do to protect and maintain the quality of the oocyte? Well, we know when we collect the oocyte from the follicular fluid, the fluid is brought through via the needle, through the tubing and into a test tube. And what I absolutely hope is that all laboratories use a hot block to hold on to their tubes and pre-warm their tubes. This, and I should have brought a prop, a hand holding a test tube is not 27 degrees, and it's important, 37 degrees, I should say, and it's important that we remember that. This is again a very old paper from 2006 from Australian group, and what they did is they actually looked at the egg collection tubing and the needle and the pathway of the egg into the tube in the hot block, and they put thermocouples along the tubing and into the tube. And what they found, that even though you use a pre-warmed tube in a hot block at 37 degrees, the temperature is maintained through the egg collection needle tubing, but significantly drops as the moment that follicular fluid hits the pre-warmed tube. Now, this is quite frightening when you think about it, because that, again, the egg is immediately being exposed. It's compromised before it's even hit the laboratory. It's being exposed to reduced temperatures of around, I think this was 28, 29 degrees. We need to remove that egg from that tube as quickly as possible and get it warmed up, based on the previous paper that you saw with eggs exposed to room temperature, for more than 2 minutes. But what the key finding from this was what we should all do is actually pre-fill our tubes, our egg collection tubes, with warmed media. And we didn't see the same drop in temperature. Now, I don't know of any laboratory that pre-warms media inside their tubes. So again, it's something that all laboratories need to consider on how best we can maintain the quality of the oocytes. So what do we need to do? Well, even if we're using a hot block, we need to map it. We need to ensure that we've looked at everywhere that tube could be located in that tube warmer, and we need to verify those temperatures. And this is just an example of a table where we've identified that we're happy with the temperatures throughout a hot block. And we do this on an annual basis. What we also need to consider is when we pour the follicular fluid into a dish and try and locate the egg. Well, at that point, we have various locations on the heated stage. And each of those needs to be mapped for temperatures to ensure that, again, we're maintaining the safe temperature for the egg. What we also need to think about is, we've got position 5 there, which is where the light source is for the microscope, and that gets hot. And we need to make sure that we're not overexposing the oocyte to increased temperatures either. So it's really important that the laboratory staff is considering all of this. And again, doing these verifications on an annual basis. We then need to map that out. We need to ensure that all our temperature monitoring equipment is calibrated. But what's important is we also consider the background temperature and heat transfer. So we need to constantly look at the room temperature, because if we've got a hotter lab, the heat isn't going to transfer from the heated stage. We're therefore going to have a heated surface. So we need to control that room temperature at all times as well. And I think what this clearly demonstrates to all of us is that we cannot improve the quality of the oocyte by the time it hits the laboratory. But what we can do is hopefully maintain that quality by doing measures such as the temperature mapping and so on. And whatever that oocyte quality is, the next step is going to show us we cannot create a good quality embryo from a poor quality egg. So if we get poor quality eggs into the laboratory and maintain that quality, it's going to be very difficult, if impossible, to improve it. So thinking about the embryo, once the fertilization occurs, the embryos, as we all know, are incubated from anything from 3 to 5 days before transfer back to the uterus or freezing. Now, historically, we would always remove the embryos from the incubator, put them onto a heated stage and assess them down a microscope. And we'd look for signs of good quality development, either on day 3, which is a cleavage stage image on the left, or the blastocyst stage on day 5, which shows, a nice clear inner cell mass, a really, prominent trophectoderm. And we would grade the embryo for quality based on this. But what we were doing was we were moving the embryos from that stable, warm environment constantly and exposing them to potential drops in temperature again. So then was obviously the fantastic introduction of time lapse. And what this probably did was really the big major step into a possible improving, we were possibly at that point able to improve the quality of the oocytes we had in our, the embryos we had in our lab. And this was because time-lapse allowed us to keep the embryos inside the incubator, even if we forget morphokinetics and embryo development at the moment and purely focus on assessing the embryo for quality. Time-lapse allowed us to leave the embryos inside the incubator, taking images every 10 seconds over 5 days, leading to over 700 images of each embryo and being able to assess them remotely. So that constant, stable environment was key. And if nothing else, was a significant change to the way we worked and will have improved the quality, without a doubt, of the embryos that we create. Anything other than that was always just maintaining the quality we had, but this continuous culture system really did have an impact on quality. The vast amount of information we got from time-lapse images was then the next step. And this is where we started to really think about how we select our good quality embryos. Okay. Now, those images using time-lapse will create videos, and we never miss a thing. Now, whereas we used to take the embryos in and out of an incubator and only look at them as set timeframes, we'd miss embryonic development in between those times. Time-lapse has removed that, and we can see everything. And what we can actually see is that it's the pathway and the developmental pattern of the embryo that is just as important in how it looks. And this vast amount of data led to a significant amount of publications around algorithms. Algorithms on how we can select the embryo with the highest implantation potential, to also how we can deselect embryos and find embryos that are developing abnormally, and we can remove them from our potential cohort of those that we would transfer. So what this again ideally would allow us to select the embryo of most potential, of improved quality. It wouldn't improve the quality. Morphokinetics and algorithms were never going to improve the quality of the embryo. What it was going to do is allow us to select the embryo, and I think that's important, selecting the embryo of best quality. There was, however, a Cochrane review in 2019 that said that the evidence was still not there around morphokinetics and the use of time lapse in selecting embryos. I would say that from a personal perspective, we have used 100% time lapse. It is fantastic for deselecting our embryos, for those embryos that show abnormal development, and we know they will never be able to repair that. It's a very useful tool for that. But with building our own algorithms using morphokinetics is an extremely time-consuming and laborious process, and we are yet to and we have thousands and thousands of embryos in this algorithm. We have yet to have the strong enough evidence to fully support the use of algorithms in embryo selection. Other areas that we can look at of embryo quality and how we can select the best embryo to improve implantation potential is the genetics of the embryo, and this would be under the pre-implantation genetic testing, for example, for aneuploidy. This requires a biopsy of 4 or 5 cells from the trophectoderm, so the outer ring of cells of the embryo, and the embryo is then frozen, and those cells are sent off to a genetic testing laboratory for sequencing, and we look for signs of aneuploidy or genetic chromosomal imbalance, and look to see whether these embryos have the potential to implant. This initially was used for patients of advanced maternal age because they are more likely to have genetically imbalanced embryos, but also those with recurrent implantation failure or recurrent miscarriage. And what the genetic testing results will come back as is a euploid embryo. A euploid, it will say an embryo is euploid, which essentially means it has the correct number of chromosomes, or aneuploid, where it has the incorrect number. Or there may be the embryo is mosaic, where some of the cells were seen as normal and some are seen as abnormal. But aneuploidy is absolutely, the evidence is there to support that the embryo will arrest, it will fail to implant, it will, if it does implant, it will end up miscarrying, and we know that aneuploidy embryos should not be used for treatment. So if we don't perform PGTA, the chances are that quite often we are transferring an aneuploid embryo without knowing. And this will have a significant impact on the implantation potential of that embryo. But even though we perform PGTA, are we actually improving the implantation potential of the embryo? No, we are not. We cannot improve the implantation potential of an embryo. What we can do is look at it and see what that potential is. Has it got potential or has it not? And that will allow us to not select that embryo and potentially get to a pregnancy quicker by selecting the embryo that is euploid. And that's really important that, again, PGTA is not improving implantation potential. What it's doing is improving our selection process for selecting the embryo with increased implantation potential. There is a wealth of evidence to support PGTA and the positive outcomes and improved outcomes for our patients. However, again, if we go to our UK regulator, the HFEA, they have deemed PGTA as a red in their traffic light system, saying that it has, there's no evidence that it will improve the chances of success. And that is potentially because if we look at it per cycle started, so when a patient starts their cycle and then has PGTA, they're less likely to have an embryo transfer if obviously all of their embryos come back as aneuploid and cannot be used in treatment. So if you look at it on a per cycle started basis, PGTA will have a poorer outcome. If you look at it per embryo transferred basis, PGTA will give a significantly improved outcome. So again, it's important about how we look at the data. So let's think about how else we can look at genetics. There are emerging technologies where we can look at selecting the best embryo, genetic quality embryo, by a non-invasive genetic testing, where we use the culture media, the spent culture media that the embryo has been cultured in. We can take the embryo will shed DNA into the culture media, and that culture media can be tested to see if the embryo is again aneuploid, euploid, or mosaic. It's potentially less cost, it's less invasive for the embryo. However, there are limitations in that there is the risk that the embryo will always discard genetic material that is abnormal. Not always, I should say, it often will. It will sort of just extrude a cell that it knows is abnormal and cannot be repaired. And if that is the genetic material that we are then testing, is there a higher chance of showing an aneuploid embryo when it is in fact euploid because it has corrected itself? There is also the omics. So there are increasing number of metabolomic studies, proteomics and transcriptomics and so on, where we can look at actually not just the development of the embryo, not just look at the genetics of the embryo, but if we combine the genetics with how the embryo metabolizes, then we've got an even stronger picture to show that embryo of increased implantation potential and that embryo of better quality, that we can select that embryo to give a better chance of success for the patient. Again, pointing out that we can never improve that. We can never improve the embryo quality, but what we can do is select the one of improved quality, if that makes sense. Increased quality, I should say. There is now more and more emerging technologies for embryo selection around artificial intelligence. All the morphokinetic data has a human element attached to it, and we are often required to make decisions on when the cell divides from 1 to 2 to 3 to 4 and so on. And time-lapse will always have that subjective side to it. Artificial intelligence will use deep learning algorithms to constantly learn and respond to how the embryo develops and to the clinical outcomes. And it will almost mimic sort of human brain networks to constantly build and rebuild algorithms to help select embryos. Again, I would argue we have tested one AI system in our laboratory against our own algorithm, and have found that in a small group of selected embryos, neither of them correlates to implantation success. So just to summarize, we need to think about can or if we can improve oocyte competency and quality. Can we do that before they reach the laboratory? The data is scarce. The data is poor. We don't know if we can or we cannot, but what we need to do is ensure our patients are fully informed and consented and understand the impact of some of the potential supplements they potentially want to take. Once we have an egg entering the laboratory of whatever competency level it's at, how can we maintain it? Well, we can maintain that competency of the oocyte by being extremely careful with our laboratory procedures, with our temperature monitoring, with how long we manipulate our oocytes for outside of the incubator. And there's so much we can all go back and look at our own laboratories and question that. And finally, can we improve embryo quality and its chance of implantation? No. But what we can do is constantly look at improving our selection methods and ensuring that the patient can have the embryo of the highest potential for implantation replaced first, in the hope that their time to pregnancy is shortened. This just leaves me to say thank you very much for listening. I'm sorry I have run over, and I'm happy to answer any questions. Thank you.